Welcome to this video on glycolysis, where we will explore the biochemical pathway that breaks down glucose to produce energy, a crucial process for all living cells. Glycolysis is also known as the Embden-Meyerhof-Parnasse pathway in honor of the major contributors to its discovery and understanding: Otto Meyerhof, Emil von Emden and Jakob Parnasse. It is a 10 enzymatic steps that occurs in the cytoplasm and breaks down glucose, a 6-carbon sugar molecule, into 2 3-carbon molecules of pyruvate. Let's look at the overall equation of glycolysis. The pathway involves 1 molecule of glucose, 2 molecules of nicotinamide adenine dinucleotide plus, 2 molecules of adenosine diphosphate, and 2 inorganic phosphate ions. These react to produce 2 molecules of pyruvate, 2 molecules of NADH, 2 molecules of ADP, 2 molecules of water, HO, and 2 hydrogen ions. It involves 10 enzymes, each playing a vital role. Hexokinase, or glucokinase in the liver. Phosphohexosisomerase. Phosphofructokinase 1, PFK1. Aldolase. Triose phosphate isomerase. Glyceraldehyde 3-phosphate dehydrogenase. Phosphoglycerate kinase. Phosphoglycerate mutase. Enolase. Pyruvate kinase. Glycolysis has 2 phases, the investment phase and the payoff phase. Let's talk about the investment phase. The first step involves the enzyme hexokinase, or glucokinase in liver cells, which catalyzes the conversion of glucose to glucose 6-phosphate. This reaction uses 1 molecule of ADP to transfer a phosphate group to the glucose molecule. The next step in glycolysis is the conversion of glucose 6-phosphate into fructose 6-phosphate. This step is catalyzed by the enzyme phosphohexosisomerase, also called glucose 6-phosphate isomerase. The enzyme facilitates the isomerization of glucose 6-phosphate, an aldose, into fructose 6-phosphate, a ketose, which is necessary for the next stage in glycolysis. After the conversion of glucose 6-phosphate to fructose 6-phosphate, is the phosphorylation of fructose 6-phosphate to form fructose 1,6-bisphosphate. This reaction is catalyzed by the enzyme phosphofructokinase 1, PFK1, which is a key regulatory enzyme in glycolysis. The enzyme uses 1 molecule of ADP to add a second phosphate group to the fructose 6-phosphate molecule, converting it into fructose 1,6-bisphosphate. The next step after the formation of fructose 1,6-bisphosphate in glycolysis is its cleavage into 2 3-carbon molecules dihydroxyacetone phosphate, DHAP, which is a ketose, and glyceraldehyde 3-phosphate, G3P, which is an aldose. This reaction is catalyzed by the enzyme aldolase. After fructose 1,6-bisphosphate is cleaved into dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P, by the enzyme aldolase, the DHAP is converted into G3P by the enzyme triose phosphate isomerase. This step ensures that both products from the aldolase reaction are in the G3P form, which is the molecule that continues through the rest of the glycolytic pathway. So after this step, 2 molecules of G3P are available to continue through glycolysis. The energy payoff phase, or energy generation phase, of glycolysis begins with the conversion of glyceraldehyde 3-phosphate, G3P, to 1,3-bisphosphoglycerate, 1,3-BPG. G3P is oxidized in the presence of NAD and an inorganic phosphate to produce 1,3-BPG. The aldehyde group of glyceraldehyde 3-phosphate, G3P, is oxidized, loses electrons, by NAD+, reducing NAD and to NADH. At the same time, an inorganic phosphate, Pi, is added to G3P, resulting in the formation of 1,3-bisphosphoglycerate, 1,3-BPG. This step is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. 1,3-bisphosphoglycerate will then turn into 3-phosphoglycerate with the help of phosphoglycerate kinase, along with the production of the first ADP molecule from glycolysis. 3-phosphoglycerate will then be converted, with the help of phosphoglycerate mutase, into 2-phosphoglycerate. Two-phosphoglycerate is dehydrated to form phosphenol pyruvate, PEP, by the enzyme enolase with the release of H2O. Due to the unstable state of PEP, pyruvate kinase will facilitate its loss of a phosphate group to create the second ADP in glycolysis. Thus, PEP will then undergo conversion to pyruvate. This step is catalyzed by the enzyme pyruvate kinase. The standard Gibbs free energy delta G naught prime for the hexokinase or glucokinase reaction is -16.7. The standard Gibbs free energy delta G naught prime for the phosphohexose isomerase reaction is +1.7. The standard Gibbs free energy delta G naught prime for the phosphofructokinase 1, PFK1, reaction is -14.2. The standard Gibbs free energy delta G naught prime for the aldolase reaction is +24.3. Delta G naught prime for triose phosphate isomerase reaction is +7.5. The standard Gibbs free energy delta G naught prime for the glyceraldehyde 3-phosphate dehydrogenase reaction is +6.3. The standard Gibbs free energy delta G naught prime for the phosphoglycerate kinase reaction is -18.5. The standard Gibbs free energy delta G naught prime for the phosphoglycerate mutase reaction is +4.4. The standard Gibbs free energy delta G naught prime for the enolase reaction is +1.8. Finally, the standard Gibbs free energy delta G naught prime for the pyruvate kinase reaction -31.4. Glycolysis is highly relevant in biology and biochemistry due to its critical role in cellular energy metabolism. Here are several key reasons why glycolysis remains important. It produces high-energy molecules. In fact, glycolysis converts one molecule of glucose into two molecules of pyruvate, producing a net gain of 2 ADP, adenosine triphosphate, molecules, a direct source of energy, which is used for immediate cellular work. 2-NADH, nicotinamide adenine dinucleotide, molecules per glucose molecule. NADH is an essential redox factor primarily responsible for transferring electrons from the tricarboxylic acid, TCA, cycle to the electron transport chain to produce energy in the form of ADP. Let's talk about anaerobic and aerobic metabolism. Glycolysis does not require oxygen and can function under anaerobic conditions. This makes it crucial for cells that lack mitochondria or for environments where oxygen is scarce, such as in muscle cells during intense exercise. Under anaerobic conditions, the end product, pyruvate, is converted into lactate, in animals, or ethanol and carbon dioxide, in yeast and some plants. In the presence of oxygen, glycolysis is the first step that leads into the more energy-efficient aerobic pathways, such as the Krebs cycle, citric acid cycle, and oxidative phosphorylation in the mitochondria. In addition to glucose catabolism and energy production, glycolysis produces several relevant molecules essential for cell sustenance. For example, glucose 6-phosphate can enter the pentose phosphate pathway, producing several pentose sugars and NADP for cholesterol and fatty acid synthesis. G6P is a starting point for glycogen synthesis. Fructose 6-phosphate, F6P, can enter the hexosamine biosynthetic pathway. G3P can be converted into glycerol, essential for production of triglycerides and phospholipids. 3-phosphoglycerate, 3PG, can enter the serine biosynthesis pathway. Phosphenol pyruvate, PEP, can enter the aromatic amino acid biosynthesis. Pyruvate, which has multiple pathways, synthesis of alanine, oxalostate, and acetyl-CoA. Thank you. If you enjoyed this video and want to see more content like this, don't forget to hit that subscribe button. It really helps support the channel, and I'd love to have you join the community. Thank you so much for watching. Your support means a lot to me.